I guess I'm back to the normal room now. And now I think I can share it.
Okay. Okay, it's ready.
Is it working now? Can you see my slides?
I can see your presentation, doctor.
Okay, so that's good. So let's go on. Again, let me thank you all for the kind invitation. It's a real pleasure to be here, actually to celebrate with you this 50th anniversary of the society. So it's an honor to be here. And my idea here is to pinpoint some things that we have done in this field. So trying to select some examples where we apply the tools that we have been developing, and also trying to show some, let's say, some systems that can help us with this big understanding. So the goal for today, I mean, the plan that I have is to start with a brief, at the same time, trying to bring into the discussion points that are often neglecting in most of the bioinformatic applications, and they are really important. So is the pH effect. And this aspect, I mean, trying to recover some points, I mean, some approach using the framework that we have more in physical chemistry. Then I go to the models that we have been developing and applying for proteins, polyelectrolytes, nucleic acids. Then again, some examples of applications that we can see some, let's say, for us at least, nice things, and trying to show you directions that we are going right now. So this is, let's say, the goal for this 40 minutes of talk that I have with you. At the same time, I know that there are students, so it's important to remind you that bioinformatics is a very broad, and it's in some way is an independent discipline, but it's all connected with all the subjects. And when I say so, I'm telling you that, of course, it can guide innovations, so we can apply this to biotechnological things. But it's important to know from what kind of a background I'm coming from, and not going to be my, let's say, my approach, I mean, which eyes I'm using to look to bioinformatics. So I remind you that I'm coming from physics. So when I'm walking in this bioinformatic path, I'm bringing tools from physics. So you can see in this slide that I'm talking about pH, and I mentioned this peculiar physics. So I'm going to bring this physical chemical approach in my descriptions. And to help with understanding what we are doing, I guess it's nice to remind one of the, let's say, there are several definitions of bioinformatics. And one of my favorite ones is from Mark Gerstein, where he can highlight things that what we are doing in bioinformatics is to understand biology. I can also say medicine in general. I mean, we can expand that we are looking and trying to understand for life in a very broad definition, but in the terms of molecules. So in this sense, I mean, more in this structural bioinformatics, so let's say subdivision of bioinformatics, and using physical chemistry approach, as you can see here in the screen. So this is, for me, one thing that's very important to highlight, because often we are talking about bioinformatics with so different approach that people can get confused exactly what we are really doing. So this is something important. And I think the key difference between computational chemistry and bioinformatics, or computational biophysics and bioinformatics, is that often we are trying to run calculations on a large scale. So either we repeat for many different systems or many different physical chemical conditions to explore these things. So this is something, I mean, to put this in the framework, what we are doing in our lab. So now from the, let's say, this big picture, I'm starting going narrowing down, and I'm trying just to tell you what we, in fact, we do in our lab. The main goal is to understand the biomolecular interactions. Could it be the interaction of a protein with a protein nanoparticles, protein polyelectrolyte, protein nucleic acid, whatever we do in terms of biomolecular interactions. And to do so, we develop and apply models. So we write our own computational tools, or we use computational tools that are available, or we are between both ways and we try to modify available tools. A few examples about tools that we have been developed is FPTS, is a titration scheme for constant pH simulations, and the proceed pKa, where we apply these ideas of physical chemistry to identify epitopes in protein related to virus. So this is things that we have been developing. For instance, we also use available tools like hygromics for molecular dynamics. And our goal is, of course, to understand the molecular basis of nature. One of our favorite examples during the last 10 years has been virus and therapeutic binders, but not just for therapeutic binders, not just for therapy, but also for diagnosis and prevention. And I think the whole idea here is that we can use this combination of approaches, is particularly focused on computational aspects, to try to understand a little bit better nature and eventually to develop new applications. So this is, let's say, again, I'm narrowing down what we do to try to give a better or clear example about our tools or our approach in general, how we use them to rationalize more complex systems. I think I can give an example here about what we use in microencapsulation, for instance. If you have a drug that's very sensitive to the environment, you have it to protect in a kind of a capsid, and in a very specific condition, you need to release this drug. So this is a complex problem. What we try is to identify the key elements here. And I think the key elements here is to understand that to form this capsid, we need to understand the interaction between a polyelectrolyte and a protein. And in order to understand these key elements, we need to build up a model. And in this case, we know that pH and salt concentration is really very important for this system. So we build up a model that's a kind of toy-like model that allows us to study the system and to measure the properties that we are interested in. So using this kind of approach, we can measure, for instance, titration plots. As you can see here, I have the charges as a function of pH here. So this is a titration plot. We can also have here examples of the free energies of interactions. So to bind the binding affinities under different conditions, we can get properties like virial coefficients that are very useful in thermodynamic studies. And the same thing I can apply for any kind of systems that would like to study biomolecular interactions. So we can easily change now to something even more complex. That's the biological system. Where would you like to study immunoresponsible, for instance? Would you like to study how the virological aspects of any virus? I mean, we can explore different things, but it's the same approach. I mean, from this very complex system, we identify key elements. For instance, in this case, an antigen and an antibody. We do again the same kind of approach to build up a toy model, and we do this to study again pH, salt, and mutation effects. And doing so, we can get all the properties that we have been discussing so far. We try to have models that are fast. The reason is we need to repeat this calculation for different conditions, different pH, different salt, and different mutations. So molecular dynamics, for instance, is great, but it's going to be a very slow process if you like to try as many conditions as possible. So in this case, we need to think about what we can do to simplify the models to get something that's faster. And again, using such tools, we can get properties very similar to what I just described to you for the other system, charges a function of pH, we can get the free energies of interactions. And in the case of the antigens and antibodies, we are, of course, interested in epitopes and paratopes, both at the sequence level and at the surface level. So this is, I mean, as you can see, using this approach to build up models specifically to address one question, we can generalize the approach. And as soon as we are talking about the interaction of a pair of molecules, we can adapt it to different systems. So once we learn about the context of the problem, you start be able to go further and make progress in your, let's say, estimation of the quantities. And most of the things that can be quantified in these models, they can be directly compared with experimental data. So this is also nice because you can validate your models at the same time that you can go further and make predictions. So now that I have introduced you what we are doing, I mean, this is the modeling part. Let me just have a flash introduction about pH. I mean, I mentioned to you that pH is very important, and you can see that's going to be even more important for biomolecular interactions. So we often know that pH is related to the concentration of the hydrogen. But what we often forget is that pH is controlling the charge of your system. So the charge of your system is a function of pH. And if you were changing the charge, you were changing the electrostatic interactions. And if you change the electrostatic interactions, you can change a lot the binding affinity. So there are many examples where the biological function is a function of the pH. So I'm just showing here classical examples from textbook for 3 different enzymes, and you can see that their activity is a function of pH. So different values, you have the maximum in different pH conditions. But we also know that the charge changes, also the conformation of the molecule. So you can fold and unfolding a molecule, change the pH conditions. And when you change the pH conditions to open a molecule, to open the conformation, you are also exposing more amino acids that are supposed to be in the core, and they have a tendency to be more hydrophobic. So you can also increase other, let's say, interactions that can be in some way enhancing what you have for the charge mechanism. So you can see this for many different examples in terms of association with proteins. This is very much used and explored in the food industry, in the beer production, to deliver drugs, for instance, or to prepare soft drinks or energetic drinks, sport drinks, these kind of things. So this is just a couple of examples. But what we often forget, and that's where I bring the physics, is that if you go back to old studies, I'm not talking about something that has just been taken from the old one, but now I'm going back to 52, when Kirkwood and Schumacher, they have presented this very nice paper where they have analytically demonstrate that even in the condition that the 2 molecules, they carry the same net charge or the same sign like charges, they can form a complex. Come on, how on earth 2 molecules that they carry the same net charge, they can form a complex? And they have demonstrated that this is due to this term here that's related to what we call charge fluctuation. So the molecules, they have a property that in the presence of another charged objects, their chemical equilibrium for the acid and base is going to be shifted, and this gives rise to an attraction. I'm not going to all the details here. I'm happy to take questions later, but just to tell you that this has been demonstrated in terms of statistical mechanics, and it has also been confirmed in experiments. So all the time we have a term that is going to be repulsive if the 2 contributions are coming from like charge, but all the time we also have a term that can be attractive. And eventually there are conditions that the green part of this equation that I'm highlighting for the screen can overcome the red part, and then we have a domination of this charge fluctuation mechanism. In terms of experiments, let me just show one of them. I mean, there are many of them, just one classical example where we have the complex association or complex conservation between whey proteins and gamma arabic. And when they are under this condition here, very close to the isolectic point, where the net charge of one molecule is 0 and the other one is still charged, they form complex. But they can also form a complex a little bit far away from the isolectic point, where they do have like charges. So this is a kind of a paradox. Come on, I'm telling you that a negative protein and a negative protein can associate. It's a kind of crazy things. I mean, if you go back to what you learned from the basic school in terms of Coulomb law, this is going to be totally insane. But we have seen several experimental data, I mean, from the lab, it's not from the computers, that they can show you that this is happening. So we have the theory from Kirkwood and Schumacher proving that this can be possible. We have lots of experiments proving that this can happen. So now we have to discuss why this is happening. And to make the things a little bit more complicated, we are talking about salt that's monovalent. And it was new in the community that if we have molecules that they were under trivalent or high valency salts, we could have a phenomena called ion correlation. But this is not true. I mean, we have, it is not the case. We have a situation that all the previous theories would not work, except if you take into account pH in the game. In terms of simulation, we have done a couple of experiments to demonstrate such things. For instance, we have taken the 3 milk proteins that were studying most of the experimental setups, and we can see here that we can find the complexation. I mean, showing you on the screen the free energy of interaction as a function of separation distance. And the only way to see it is if you have charge fluctuation, if you have the pH effect. If you perform the simulations using a constant charge approach, even if at the very beginning of the simulation, you set up the charge to be the precise one, you end up into troubles. You're not going to get the complexation. We have been trying different things also to check if this was something specific or non-specific interactions. And we can see if you map where, for instance, the other molecule is exploring from the surface for the target one, we can see shifts here as shown by these different colors happening all over the surface. Whatever you see a color here means that it has been visited by the other molecule. So it's something that's not specific, I mean, it's no specific interaction, but can be quite strong to give you a complexation. So this is to illustrate to you that there are many more, let's say, conditions that we often, when you're talking about in bioinformatics, we don't take into account if we don't explore the physical aspects. So my point here is just to highlight to you very briefly some ideas where the fact that you can use physical chemical approaches help you to be much more realistic in what you were modeling in bioinformatics. So combining these things is what we often do. So our modeling is to try to explore as much as possible electrostatic interactions. And not just as much as possible, as complete as possible. And we have assumed that most of the systems that have been working at least are the ones that electrostatic interactions are the main driving force. We need models that should describe pH, salt effect, and mutation effects. They need to quantify binding affinities. We need the free energy as a function of the separation distance of the molecules. So this goes far away from just exploring what we do in docking studies, that we just explore the contact surface. Now we are exploring the whole range, how they approach each other. What is the driving force for them to form a complex? And we also need to repeat this calculation in different conditions. So we needed to put everything together to be able to describe a reasonable decision that we want in a reasonable CPU time as well. And we have to remember that we are in Latin America, so we have a small budget. So we have not the same computational resources that people have in other places. So we needed to think about all these constraints to build up our models. And it's the direction that we have started developing our tools, making this balance between how much we can simplify to reduce the CPU costs, at the same time keeping it realistic, to put the physics that we need in the model to be realistic and to provide accuracy and to be, of course, of a practical relevance. So from a given call, I mean, remember in physics, we like these things, I mean, spherical buzzer or spherical call, but from a given call, there are different ways to represent it. And it's just a matter to define what level of resolution we need in our model. Sometimes it's enough to go to this level here. Sometimes we need more details. So again, the good model is dependent what questions you were trying to address and what resource do you have to address such questions. So there is no ideal model for everything. There is an ideal model for each application, each question that you are trying to address. So keep this in mind. It is very important, in my opinion, at least. And just to show you a couple of examples about models that we have been developing, we have started with a very, very coarse definition using colloidal-like models, like just to say that a protein is a spheric object with a different charge distributions. Could it be double, could be one, could be a combination. This is what we call colloidal-like models. Then we have evolved to put an atomistic description, confine everybody in a cell box, and be able to couple it with a proton box. So we could introduce the protonation states. I mean, we could change the protonation state of the molecules using the pH as an input parameter. So we have in a normal simulation, we have a temperature, we have a pressure that can be input parameters. In our simulations, pH is also one of the input parameters. And we have been exploring it in different directions, using explicit and implicit ions. So one, let's say, model that has been very successful, particularly because it decreased a lot of the CPU time and allows us to run longer and longer runs. So convergence is not an issue any longer, is the FPTS, the fast proton titration scheme. We have applied for protein-protein interactions, for instance. That's what we have our fast coarse-grained protein-protein model. And I mean, combination of such things as ProCD-pKa, and also trying to couple to molecular dynamics in what we call PEP6, that we have a couple between titration and conformational changes. And we keep going in all the directions. I mean, I'm not going to have time for all the details, but just to give you an idea, I mean, one problem that we have, this actually was the motivation to build up FPTS, was that when we try to do simulation with explicit salts, and we try to do Monte Carlo movements, try to move our particles, it was very hard to make movements, because if you have explicit ions, there were overlaps all the time. I mean, you try to move your big particles, your big proteins, and they end up stepping one of these small contrarian objects. So the convergence was really a critical issue. We could easily solve it, just get rid of the explicit description and make it implicit salt description. But then you can ask yourself, come on, you were simplifying too much. How realistic is this to it? So, well, we have started benchmarking it. We have started making comparisons between our approach and different data. So, for instance, we took here the 3 proteins that are very often used to benchmark the quality of electrostatic models. And in this case, we have seen that the experimental data, we have here the comparison with the generalized Born approach. We have all atom constant pH simulations using molecular dynamics, and we have our FPTS. If you use the basic descriptors, like the maximum absolute deviations or the average RMSES for these guys, we can see that our approach is very, very close to what you can get for all the others. And actually, it's a bit more, I would say, is slightly better than even all atom simulations. But we pay a much cheaper price to run the calculations. Most of the models, they have troubles to reproduce one given amino acid, this glutarate-5 from lysozyme. You can see that some models, they overestimate it, others underestimate it. But at the end of the day, they all have troubles with just one specific one. I mean, I can show you different benchmarks, but let me just go back, because I was so quicker that eventually you didn't pay attention what's the condition of our modeling. In this simplification, what we are using in our Hamiltonian is a Coulombic contribution plus a contribution here that's related to the net charge of the protein and what's happening in the solution. This kappa here is the Biron length, is the Biohookel length. And the beta here is not the Biron, but is the size of your protein. So what we do is we try to move one proton to the solution from the protein, and we recalculate ZP, and we change kappa. And applying a Monte Carlo criterion, we define if you're going to accept or not this movement. And then we go back. I mean, we can also go see if the process on the other direction can also happen. I mean, this is random selected. So essentially, you pick up random titratable groups and try to protonate or deprotonate using this equation. So this is all the trick that is given in the model. Just, it's very simple and it's very efficient. As you can see here in this comparison, we get reasonable results. There are different benchmarks. I mean, we can continue here selecting different proteins. Of course, how good is the model based in some selected case can be biased. So for the system that we have been studied, there are systems that we are better, there are systems that we are equivalent, and eventually there might be other systems that we haven't studied that we can be even better or even worse. I mean, but at least we are able to capture the main points of most of the models. I'm not selling FFTS as the best model available, but I'm selling FFTS as one good choice if you like to decrease your CPU time and have reasonable results. So this is the main, let's say, take-home message from these results. For instance, in this comparison here with another approach that's available that's much more computational costly, you can see that our results are much better. But eventually there are other systems that we are not going to do so well. I took a comparison here with PropKA, for instance. PropKA is very fast. Essentially, you put your PDB there and you press the button and you get the results. So this gives you a comparison, how fast you can go for an approach that's not going to incorporate all the physical features because you don't specify, for instance, the salt concentration in PropKA. And in FFTS, you can specify. In this case, we can see that if I compare the experimental data with the theoretical results, we have a very nice correlation compared to what you can get for PropKA. It's slightly better. Both methods have troubles for some specific amino acids. As you can see, there are points very outside what you are going to expect then. But again, for this set of proteins that were studied here, we are doing a reasonable job. With a great advantage. If I compare, for instance, CPU costs, I can compare with Poisson-Boltzmann and FFTS. We are at least 2 orders of magnitude cheaper for a very large protein than Poisson-Boltzmann. So this is also something very positive. I'm jumping a few slides. I mean, we have done comparisons with nucleic acid as well. But one good point here to highlight is that convergence. I mean, our simulations we don't have this convergence problem as other approach they have seen. I mean, you can see that it takes about 40 nanoseconds for some systems to converge in different approaches. And in other case, can be a little bit less, but typically convergence is an issue. In our case, in just a couple Monte Carlo steps, we have it converted. And if you look in terms of CPU time, it takes about fractions of minutes. So this is something very positive. We have been improving it because we are not satisfied with the results. So we think that there's still room for improvements. So one thing that we have done is to use an old idea from Antonio Batista, where they have a couple GROMACS, molecular dynamics simulation with a Poisson-Boltzmann solver called MIAD. So we took this idea, and the whole point here is that you run a molecular dynamics, and from time to time, you call a titration scheme. In their case, they call this Poisson-Boltzmann solver. In our case, we have adapted it to call FPTS. And instead of use an atomistic force field, we have used OPEP. OPEP has an advantage because it has been designed to work with the continuous solvent, implicit solvent. It has been very successful to predict folded structures of peptides. And furthermore, we have this additional point here that's very important. It has already prepared because it has already used ion pairs. So essentially, the titration scheme can define if I'm going to switch on or switch off an ion pair. So OPEP had already the structure that to make this coupling would be very appealing. And we have done so, and we have done a comparison. It's surprising to see that what we gain with OPEP, repeating the same studies that we have done for other proteins before, is not really a big improvement, but it's a slight improvement. I mean, you can see, for instance, that we have, let's say, a maximum deviation here of 0.9 if I'm using the rigid model. But if we go for OPEP here, we go to 0.7. I mean, we gain something, but we pay a much higher CPU time. So depending on what we are doing, it's not really appealing to pay more to get essentially the same results. So that's why very often we are still using FFTS and not OPEP 6 because we see that it's much more convenient to get reasonable numbers with FFTS. But again, there is a slight improvement. I'm jumping because it's just a repetition of the slides and data, Now let's go to some applications. One of my favorite cases is spidering. Spidering is a spider silk. So let's apply it now, these tools for complexation studies. And one problem here was spidering is very, let's say, important in biotech. I mean, you have the biosteel, but it's a protein that's controlled by pH. So inside a spider silk, inside a spider, this protein exists as monomers. So it's not really a protein, it's just a combination of monomers. But as you start putting pH, decreasing the pH, you start putting acid, sorry, increasing the pH, you see an association between the N-terminal of this molecule. So from the experiments, we know from the, let's say, from the animal experiments, it's known that inside this kind of box here that you have in a spider, pH is around the physiological, and when the spider is going to spill the silk, it has a proton buffer here, so you get an acid, and then they form this very strong material. So it's very appealing for medical surgery, for instance. Often when you have an inflammation, pH is low, and then when the patient is recovered, this is going to go back to monomers, and it's biodegradable, so it's going to be removed from the body with not a big issue. So it's very appealing for biological applications, being so strong and so much controlled by pH. The point is to understand what was happening. It's known that in pH 7, they are at monomers, and around pH 6, they already start being as a dimer. So the main question here is, what was the driving force? Can we explain it? Can we predict? Can we suggest what mutations can improve or change the pH window where I'm going to have this complexation? Because then I can tune this material for different applications. So using simulations, we have it trying, essentially our simulation, that's the fourth approach. I have two proteins, in this case, two spiderings. They can translate, they can rotate, and they can be affected by pH. So that's the movements that they are allowed to have. And again, titration scheme. So doing so, the first thing was to compare our data with available data. So which was on the literature available other studies only for the wild type. So we have compared our, let's say, PKs for the titratable groups of spidering with this other simulation data. You can see our results. They are within the typical range that we observe in benchmarks. And we have to predict all the possible changes for all the mutations that can happen in this system. So this is, okay, we can reproduce pKa's. That's good. We have a model that's reasonable for these electrostatic features. And I think, doing that, the second step was to see if we could get the right pH window where we observe a complexation. In this plot here, I have the titration, the binding affinities, and binding affinities as a function of pH. When you see the dashed lines is when they are under monomer conditions, and the solid lines are when they are under complex. And we got exactly the pH window that's observed in the experiments. So here we have the same mechanism. The next question that the simulations could help to solve is, okay, now what is the driving force? And in this case, we have switch on and switch off different interactions. And we could see that the point was the dipole interactions. Remember that in simulations, you can play God. So you can switch on and switch off things. And using so, we can define what was the driving force. I'm not going to talk about all the mutations that we have tried, but in principle, we can reproduce the same thing and change the amino acid and check the contribution of each amino acid and how we can shift the pH window for this complex, just making combination of these amino acids. My time is probably close to the end. So I was very optimistic that I could talk to you about virus, but I'm just going to flash one thing that was very nice in terms of virus, is that their interface, their protein-protein interface is richer in terms of titratable groups. So this is why they are very appealing for electrostatic interactions. and also for this kind of modeling. And also, just to back to COVID, if I go back in 2020, when we had the beginning of the pandemic, the only information that we had was that there was SARS-CoV-1 that has a receptor bind domain, and it was known to bind AC2. So this was an opportunity for simulation because we could predict the experimental data before it was available. All the systems that I have shown so far, we are trying to benchmark our models based in the experimental data that's available. Now it was an opposite situation. We had this information and we needed to predict if the receptor bind domain from SARS-CoV-2 could bind to AC2 and use the same entry mechanism. And there was no available experimental information specifically for SARS-CoV-2. So in this moment, what we have done is to try to address if it was the same entry mechanism to enter human cells, was to compare SARS-CoV-2 with SARS-CoV-1. And what we did was to use the available structural information from SARS-CoV-1, and this was before AlphaFold. So what we have done was homological modeling, get a SARS-CoV-2 structure, and use this SARS-CoV-2 structure to do a complexation study and demonstrate that indeed SARS-CoV-2 has a receptor bind domain that could bind to AC2 receptor. So this was done before experimental data was available, which was quite nice because we could challenge our models to see if we could predict what nature could happen. And in fact, this was true. I mean, we had done a couple of comparisons between experimental data, also comparing the number of amino acids that were affected. The experimental data that I'm highlight here in red came after the publication of the paper or after the submission of the paper in bioRxiv, because this was a very dynamical process that everybody was producing data. And after this theoretical paper that we have published, there were many more in the same wiki actually that were showing exactly the same thing. But it was very nice to see that this kind of modeling could pick up the things. We have tested antibodies as well at that time. And just to jump you to the very end, I mean, it was the same idea to use the same pipelines, to try optimization, this kind of things, different approach. But there is one result that I would like to show before I go to the final end. What you can see in the screen is a correlation map network diagram using the pKa's of different proteins. In this case, receptor bind domain with different mutants. And you can see here the concentration of the beta and gamma variant. You can see here the delta, and you can see the Omicron up to BA3. So this was done in 2022. This was the Omicron variant that was circulated at that time. And doing so, what we get here is exactly what you have in phylogenetic trees. So nature is exploring electrostatic features to make the receptor bind domain much more, to increase its affinity with AC2. So it's nice to see how we can study evolution, biological evolution from the virus, correlate it with electrostatics, and explore it from a computational point of view, because we are calculate such things using a computational approach. So this is very nice, I think, to illustrate what can be done in this combination between bioinformatics with physics, with biological problems. I'm sorry that I think I have passed a couple of minutes. I'm just jumping, not to showing you all the things. I was very optimistic with everything that I could present, but probably I took a little bit more time in the introduction, so I'm jumping to the end. And let's just highlight here the main points that I try to show. Even if I jump a couple of slides, I think I could show you that simplified models can help you to understand these complex mechanisms, help you to design new materials. I think the example with the spidering is a nice one. Electrostatic selective is possible. You can see at least 2 examples of it. And Nature, as in the SARS-CoV-2 problem, is really exploring these things. I thank lots of people that were involved, from students and collaborators, of course, people that support us, the taxpayers that support us from different funding agents. And to finalize, congratulations to your 50th birthday. And thank you again. I'm happy to take any question or any comment, whatever.
Whoa, great. Thank you so very much, Dr. Fernando. It's great. Doctor, I think we have some questions in the chat for you. A moment, please. Guys, if you have any questions, please leave us on our chat.
Yeah, I have one question. Doctor, thanks for your talk. The protonation is very important. I working in solar energy material, and in the interface between materials, if there are protons, they cancel the electrons from the excitation. So it's very important for us taking in account the protonation between interfaces. So what's the main difference using the method that you explain us between charge equilibration based on electronegativity? Because the method that you told us is based on titration, right?
Mm-hmm. Yeah, in our case, we are using, let's say, what we can call an effective approach. I mean, protonation, by definition, is a quantum phenomenon. So if you just try to describe it using a quantum approach, it's going to be very computational costly. So it's going to be prohibitive, especially for the size of the system, and especially because in our case, we are trying to incorporate these things for protein-protein interactions, for instance. So the number of titratable groups is quite large and can be, depending on the size of your protein, can be really very big. So we need to invoke approximations. What we're trying is to describe a quantum phenomena using a kind of a phenomenological approach. So it's slightly different from what if I understand what you were doing, because in your case, you were really going to the more fundamental point where you would describe it as using much more quantum ideas. In our case, we simplify a lot things just to describe the general phenomena. So, for instance, with this kind of modeling, you cannot capture the path how the proton is transferring. You have a proton that's here and disappears and goes somewhere else. So you see the initial state and the final state. You don't see the transition states. So we miss this information. But we miss something that is not, remember what I said about the models. Well, what you want is the pH effect in the system, not to know exactly where the proton is. So again, depend on the question that you're addressing. In your case, it's important to know more details about where the proton is, where the electron is, because you were really focused on this specific part. In our case, it's much more general and it can be much more effective, which helps a lot, by the way.
Yeah, sure, because your model is huge. Yeah, I can notice that. So it is possible to apply for formulation, because in drug formulations, you use polymers to avoid aggregations. So there are also charges in the polymers. You can use these methods to combine with those information not from residues, not for other polymers that is in the formulation.
Well, we have used it for protein polyelectrolyte interactions.
Oh.
And the idea, I mean, was really for microencapsulation. So in principle, they can be adapted to different systems. In our case, when we did the calculations for protein polyelectrolytes, for specific reason that we had. I mean, we kept the protonation states of the polymer constant, and we only changed the protein. So it was, I mean, needed for what we were trying to explore in that moment. But we have done, for instance, protein RNA interactions, and in this case, both the protein and the RNA were able to titrate. So the protonation was changed for both of molecules under different pH conditions. So they are, let's say, the idea is quite general because it's a phenomenological description. Essentially, you just have to adapt it, and there are different levels. I mean, one level, for instance, is when you have explicit ions. The other is using implicit model. So depend what I mean looks like your system, the questions, the computational resource that you have, you can define which model is going to be better for that specific thing. So in principle, it can be even, I mean, instead of going to this coarse grain description, you can go to a more detailed description incorporating the same things. I mean, you can define if you were going to apply it to a bead, that's going to be your amino acid. You can define if you're going to apply it to a specific atom, that's going to be the one that actually is where the proton is going or not. So it's the ideas is quite general. That's why I try to flash you with these different examples, because if I could just focus on one particular problem, probably I was not going to give you this broad view about how these things can be really generalized.
It's a good tool. Thank you, Dr. Fernando.
Oh, my pleasure.
Yes.
Thank you, Dr. Fernando. Any other questions? Gustavo Livos, go ahead. Puedes activar tu audio.
Okay, thank you very much, Professor. Just I have a talk about great exposition. It was very interesting. Even I'm not working on this. I have never heard actually about. Well, I hear about explicit solvent, but not applying this as the way you apply this. I have two doubts. The first one is about the timescales that are needed for in the all atomistic with explicit solvent. Which timescales do you think at maybe at average is needed to observe such as event? Because you explained that you are using implicit solvent to reduce the computational time, but at the, let's say, at the full atomistic scale, which time of simulation would we need to observe such an event? And the other is, what methods are you using for free energy calculation? Because I think you present some figures about it, but I don't know if I explained and I didn't catch it, but just that two questions. Thank you very much again.
Well, thanks for your questions. Let's see. In terms of the simulation time, what is going to affect a lot is how big is your system, how in the number of titratable groups that you have in your system. So this is, I mean, two things that you have to keep in mind. And it's going to depend if you are going to calculate the properties of a single molecule, or you are going to apply it in a complexation study. I can give you, for instance, a comparison. A titration, I mean, to get the titration plot of a single molecule using FPTS, where we have a rigid description of the protein and we have implicit solvent, it takes about, let's say, 1 day you get a titration plot. One afternoon, depending on the system. And when you run OPEP6 using the same CPU power, it takes about 3 weeks. And OPEP6 is still a coarse description. So we didn't try an atomistic description because a couple of weeks is already too much just to get one replica of the system. And often we need to repeat the calculation at least for 3, 4, 5 replicates. So this means that just to get the titration plot of a single protein using OPEP6 would take us about a couple of months to have reasonable data using the same CPU power. Of course, if you have more CPU power, you can decrease the time. But I mean, I'm just making this comparison to you. On the other hand, if you try now to go for protein-protein interactions, a rigid model in about 1 week time, we get the free energies. But if you go for anatomistic descriptions, we take about half a year with the same CPU power. So this gives you a feeling, I mean, how to define the things. And your next question was about the free energy. In our case, when we have this pair of proteins that can translate and rotate, what we are doing is very basic statistical mechanics. What we do is we sample histograms. From these histograms, we get radial distribution functions. And from these radial distribution functions, we get potential of mean forces. That essentially is the free energy of interactions. So depend on how the system is close to each other. I mean, if they like to stay together or they like to be far away, you need only to increase the number of CPU time, I mean, to run more and more to more or less populate in an equal level all the beams. So the critical part here is that you need long and long runs, especially when the system have larger barriers. Alternatively, you could use metadynamics approach. For protein polyelectrolyte, we have been using what we call penalty functions. Some way we penalize some configurations. We added this in the Hamiltonian penalization function to try to let the system explore other separation distance. So if the CPU cost is too high, you can start thinking about these choices. But I often prefer to try to run longer because it's not totally clear how much we are affecting the titration properties of the system if we start using metadynamics. So if we have a chance to run the system in a, let's say, without playing all the tricks, it's our choice. So essentially, you wait more in terms of CPU time, but you are sure that you are not introducing any bias in your system, especially for the titration properties. I mean, this is something that can be critical in some systems, especially because if you were changing the protonation states and you are forced into be in anodic conditions because the molecules are close to each other more often than they were supposed to be, just because you're trying to get the free energies, we can end up into some critical, I mean, some artifacts in the simulations. That's not so easy to control. There might be ways to go to the statistical mechanics base and try to correct the results, but if you can skip these troubles, I think I feel more comfortable. I guess these were your two questions, right? Did I miss something?
Okay. Interesting. Stanislao, go ahead with your questions.
First of all, thank you very much, Professor Barroso, to be here in our meeting. The lecture was quite interesting, especially for me. I have one doubt about the potential uses of the model. Have you ever tried to use your approach in order to predict peptide aggregation, for example? Or try to predict the behavior of antibiotic peptides aggregation?
We haven't tried yet, but in principle, it's doable. I mean, I would say the approach has a better tendency to work, especially if the main driving force is going to be electrostatics. So for all the systems that we have any indication that electrostatic is the main driving force, it's probably a higher chance that this approach is going to work well. If the electrostatic is not the driving force, then probably you have to adjust the van der Waals contributions in the model, and that might involve, I mean, the need to reparameterize a couple of things to make sure that we can correct for things that are missing. When the electrostatic is a driving force, it's going to be, again, the dominant contribution. And even if you were making some, let's say, partial guessing wrongly something, it's more or less going to compensate by the features that comes from electrostatics. So if you have some evidence that your system is driven by electrostatics, I mean, have a pH effect in the aggregation, or you have a clear salt effect in the aggregation, might be a good suggestion that the model can work well.
Thank you very much.
Oh, my pleasure.
Well, thank you so very much, Dr. Fernando. We hope you will join us at our next events. Thank you.
Thank you for the kind invitation. Thanks for the opportunity to be here. And again, congratulations to your birthday.